Hello, everyone. My name is Eric Trotman. I'm a second year PhD student at the University of Florida in the Kaiser Lab. Today, I'm going to talk to you about my last 2 years of my research about parasite-induced changes to aquatic snail behavior in the presence of predation cues. This is only a cautionary tale because in my first year, I collected data and the sample size was pretty small in terms of number of snails that were infected. So we had a different idea of what was going on. But then the second year, when I collected more samples, we kind of got a bigger picture of like maybe what might be going on with the snails as they're becoming infected. So that's why. Um, okay, so moving over into just what these parasites are and how they move about in the environment. Um, Trophic transmission is one way that parasites are able to move from host to host to be able to complete their life cycle and be able to sexually reproduce. So trophic transmission is the movement of parasites from one host to the other by means of predation or by being eaten through some means in the environment. Um, so there are ways that parasites actually change the behavior of their hosts to make them trophically more easily predated upon. And some terms used to describe this were parasite behavioral modification and parasite manipulative parasites. But a broader term is parasite increased trophic transmission. And one such example of this is with encysted metacercariae that encyst in fish. When the fish then get eaten, these cercariae are able to move over into the next life cycle to get eaten, to move over the next cycle in this next host. Another example is in metamorphs or in tadpoles. The cercariae of these trematodes move in and they cause limb abnormalities, which cause them to be predated on easier. But there's also in the same trematode species, there's a non-trophic transmission stage. And this is when trematode parasites actively seek out their nest host. As you can see, they're not great swimmers, however, and they have multiple filters and barriers to like reproduce and multiply in their environment. So they actually have to be able to find the host in their environment, so they encounter filters, and they have to be able to be compatible with their host. And because not everything that they come into contact with, they are going to be able to um, reproduce in. So behavior modification can be a strategy for several modes of transmission. And my general question is, do non-tropically transmitted parasites manipulate their host to improve probability of transmission? The study system that I'm working in is Echinostoma trivolvus. So here, their definitive host lays, or not lays, when it defecates into water bodies, it lays the eggs, move out into the environment as well. The eggs then develop into Myricicidiae, which have flagella, and they frantically search for their first hosts, which are snails. And these Planarbella trivolvus are my study host. Within the body of the snail, the Meriscaridae develop into radii, which then release these cercariae, which also have a single flagella, which they can move over in the environment too. These cercariae then find their next host, their intermediate host, which can be amphibians, snails again, or fish, and then move into the definitive host, which are birds or water-bodied mammals. So my 3 study questions were to assess infection prevalence and intensity across 3 Panarbella tribolus populations in Alachua County, Florida, to quantify the differences in behavior of infected and uninfected snails, and to measure snail movement in the presence of predation cues. So marsh ramshorn snails are air-breathing aquatic snails found typically grazing on plants near the surface of the water or detritus in shallow waters. And we collected from 3 sites, Mossy Lake, Prairie Creek, and River Styx, all which had different habitat like variables and microhabitats. And that's something that I definitely really want to get into later on in my next years of research and honing in more on the microhabitat. So we sampled some snails and we collected 193 snails from River Styx, 149 from Prairie Creek, and 97 from Mossy Lake. Mossy Lake mainly because this year the vegetation was really dense and it was hard for us to collect as many snails as we would have liked. However, at Prairie Creek also, we expanded our search to different areas in Prairie Creek, and that's where the idea of microhabitat and looking at that more in depth came into play because we were realizing that we could collect snails in certain areas and they may have been more higher infected. Um, Hey, so the timeline of behavioral experiments. We collected the snails from the field. We measured their snail height, snail size. 2 days post, we looked for secarial shedding. Then right after that, we did their writing behavior assay. We looked at their phototaxis, so they were housed overnight from day 2 to 3, and then they had their predator assay on day 4. So secarial shedding consists of checking the dechlorinated water that the snails were kept in after 24-hour cycles, and the number of secaria trematoes were recorded in 3 separate droplets, 3 times. So each snail got a treatment of 3 and then were averaged. Um, the Across our 3 sites, we kind of found around the same number of infected individuals per snails collected. However, at Prairie Creek, there was a higher parasite intensity. So snails here had a higher load of parasites than at others of infected individuals. And that was significantly significant. So the writing behavior was looking at how these parasites might actually be manipulating their host. So we looked at 2 different things, the time to emergence and the time to suction. And this video kind of shows a quick example of that. So wait for it to start over. And so this is a time-lapse video also, so definitely wasn't going this fast. So that is the emergence time. Well, I did a predation attempt, but that's the emergence time right there. And then it has to orient its body to move completely over to find the bottom of the plate. And then that's the suction movement and when it moves again. Okay, we didn't find any differences between latency emergent, latency suction across the 3 sites or between infected and uninfected individuals, which are the red and the blue bars. We did find a slight difference with snail size, looking at the larger the snails were, the longer it took for them to emerge from their shell, which somewhat makes sense because it takes more energy for them to move, especially out of the water. So the second experiment was phototaxis, responsive to light versus the dark, and we checked the snail's positions either in the dark or the light in 1 hour, 3 hour, and 24-hour intervals. We found that infected snails took longer to move to settle to the same proportions as uninfected snails did into the light. But overall, they did get there after the 24-hour period. But over period 1 and period 3, the infected snails seem to be moving slower into the light. The last experiment was a predator assay where snails were positioned in the center of the arena out of water and movement into the water was recorded. Snails were allowed to disperse for 1 hour before repositioning, and the groups were all infected, and infected with uninfected snails. So with this, and this is all very preliminary data, but we found that uninfected snails in the presence of infected conspecific snails, they were more reluctant to enter the water. So they either didn't enter the water as frequently as groups with all infected individuals or groups with all uninfected individuals. Okay, so the 3 take-home messages were the first one, the infection prevalence was similar across sites and infection status was higher at Prairie Creek than other sites. And I'm hoping to get into the microhabitat more to look at these differences. Infected snails took longer to settle in a light environment than uninfected snails, and uninfected snails, when grouped with infected snails and in the presence of predation cues, moved into the water less than uninfected snails alone and infected snails. So I want to take a minute to thank everybody who helped me collect snails, my undergrad students who were, a joy to have in the field, my advisor, and for people showing me where I could collect these snails from the beginning. And then for that, thank you so much.